Alrighty. So just as a reminder, we still have the midterm exam going on. So I know a couple of you guys have already worked on and finished it, which is awesome. If you haven't yet, don't worry. You still have plenty of time. It is not due until the end of the day, Friday. So 11:59 p.m. tomorrow evening. So that means that you have the rest of the night after we log off tonight and all day on Friday to work on it and get it turned in. Like I was saying last week, it covers everything that we've talked about in this class so far up to this point. If you have any questions on it, please feel free to reach out to me. I might not be able to directly help you answer the question, but if you're having trouble understanding the question or understanding what type of answer you're supposed to be giving, that is something that I can definitely help you figure out. All right. So today we're continuing on our journey of learning more about the ways that cells divide in order to create new cells. So last week, we talked a lot about a couple different things that I'm just going to kind of zoom through here just as a quick little review. We talked about our chromosomes, which are these kind of short condensed segments of DNA. In humans, we have 23 pairs of chromosomes. Each one of these pairs is called a homologous pair or a homologous chromosome because they contain the same types of genes on each one of the pair. And you inherit one of these chromosomes from your mom and one of these chromosomes from your dad. That's why you have 2 copies of every single chromosome. So in most of the cells in your body, like 99% of the cells of your body are called somatic cells. So these would be like your skin cells, your muscle cells, your brain cells, your bone cells, your stomach cells, your intestinal cells. All of these are somatic cells. That's just normal, average, typical body cell. And these guys have what's called the diploid number of chromosomes. So in our somatic cells, like I was saying, we have 2 copies of every single chromosomes. So 2 copies of 1, 2 copies of number 2, 2 copies of number 3, 2 copies of number 4, and so on and so forth. Because we have those 2 copies, we call that a diploid cell. So diploid for 2, meaning 2 copies of every chromosome. Certain cells called our gametes, so these are our sperm cells in males or our egg cells in females. These are the sex cells of our body. These guys do not have the diploid number. They have what's called the haploid number. So they have just a single copy of each chromosome. And we're going to talk a lot more about gametes and haploid cells in our class tonight. So diploid cell is our normal somatic cell, 2 copies of every chromosome. Gametes, sex cell, haploid cell, only 1 copy of every chromosome. So in humans, our haploid number, our single chromosome number is 23. So we have 23 different chromosomes. And in our normal cells, our diploid cells, that means that we have a total of 46 chromosomes in those cells. 23 pairs gives us 46 total in our diploid cells. So last week we talked about, or not last week, on Tuesday, we talked about the cell cycle and how a lot of a cell's lifetime is spent kind of growing and developing. So we have the G1 phase and the G2 phase, which are sort of like growth and development phases. If we're going to divide the cell soon, it'll go through the S phase, which is where our DNA gets copied over. All three of these guys are part of the overall process called interphase, which is sort of like the in-between phase of splitting one cell to splitting the next cell. Once we're all grown, copied our DNA and grown some again, then we very quickly go through mitosis, which is cell division, like normal everyday cell division. Go through cytokinesis, where we do the final split of the cell, and we end up with two brand new cells that then both start going through that long G1 process all over again. So this is like the life cycle of the cell. So we call it the cell cycle. We talked about the different stages of mitosis, which are all kind of nicely summed up in this picture here. So we start off with our original parent cell. We have a nucleus that's surrounding our chromosomes. We then, if we're going through mitosis, we're finished up with all the growth and development. We're finished up with interphase. We're moving into that short mitosis phase. We go through 4 different stages. Prophase, where the nucleus kind of disappears and the chromosomes start to get attached to the spindle fibers, these little kind of threads going through the cell. In metaphase, which is like the middle phase, the spindles cause the chromosomes to line up down the equator of the cell. So they line up right up and down the middle of the cell. In anaphase, that's like the action phase is what I like to call it. This is where those little spindle fibers pull the chromosomes. They split the chromosomes apart. Because remember, earlier on, we made duplicates of all of our DNA. Right? In the S phase, we went through DNA replication, made completely new set of DNA. And so in anaphase, we take those 2 sets of DNA and split them onto either side of the cell. We then move into telophase, which is kind of like the final stage of mitosis. The chromosomes have been separated, so now the nucleus starts to reform around them. They start to become a little more spread out, a little less condensed. And eventually, these 2 cells will split apart from each other and become 2 separate, brand new, what are called daughter cells. So when we're talking about cell division, a lot of times scientists refer to the original cell as the parent cell and the newly created cell as the daughter cells. So the big thing, the most important thing to remember about mitosis, if you remember nothing else, remember this. At the end of mitosis, we get 2 identical daughter cells that are diploid. So each of these cells has a full set of 46 chromosomes, and each of them looks identical to not only each other, but also to the original parent cell. So they have the exact same set of DNA as the parent cell, the exact same set of structures and organelles. They're going to do the exact same job as the parent cell. And because they have the exact same set of DNA, that means that we have diploid cells. They inherited all of those copies of each set of chromosomes. So we make duplicates, we line up the chromosomes, we pull and split them apart and form them into 2 new identical cells. Same exact DNA, same exact structure as the original cell. that is what happens in mitosis. Mitosis is like the normal, average, everyday, happens all the time type of cell division. All right, so I know that was a pretty quick run through of everything that we talked about on Tuesday. But does anybody have any questions about any of that? Anything about mitosis you want me to go over again really quickly? All righty. If not, then we shall move along to the next type of cell division, which is meiosis. So probably the worst thing about cell division is that mitosis and meiosis sound so similar, and they do similar jobs, but meiosis is like a very specialized version of mitosis. So this doesn't happen in just a normal everyday cell. Meiosis is special. Meiosis is different. So meiosis is the cell division that produces our gametes. So our sperm cells and our egg cells don't come through regular mitosis cell division, unlike all the other cells in your body. So all of your skin cells, all of your bone cells, all of your muscle cells, all of your brain cells, they are all here due to mitosis. Only your sex cells, your gametes, are here because of meiosis. So as you might expect, meiosis occurs in the places where you'd find these sex cells. So in males, it occurs in the testes, and in females, it occurs in the ovaries. And the reason that meiosis is different is that you start off with a diploid cell, a normal kind of cell, just like you would in mitosis, except by the end of meiosis, you have 4 new cells, and these 4 new cells are haploid. So you remember I mentioned earlier that meiosis produces haploid cells, or not meiosis, but your gametes are haploid cells. So at the end of meiosis, all the cells that have been produced only have 1 single copy of every chromosome. Not 2 like your normal cells, just 1. That's what it means to be haploid, and that's what happens to your cell at the end of meiosis. So there is a very particular reason why meiosis produces these gametes that only have 1 set of chromosomes. So as I mentioned earlier, meiosis occurs in order to produce our sex cells. So it produces sperm in the testes and eggs in the ovaries. And then when an egg gets fertilized, we have to combine all of the DNA from the egg and the sperm together, right? We don't just get rid of 1 set of DNA, because in a normal diploid cell, like most cells of your body, we have 2 copies of every chromosome. And who remembers where we get each one of those copies from?
From our parents.
Exactly, from your parents. You get 1 copy from your mom and 1 copy from your dad. The reason why you only get 1 copy from each parent, even though the cells of each parent have 2 copies, is because your sperm cell has 1 copy of each chromosome. The egg cell has 1 copy of each chromosome. So when the sperm and the egg join together, that's when you get 2 copies of each chromosome. So your dad's chromosomes are in the sperm, your mom's chromosomes are in the egg, and when they meet up and fertilize, that's when you end up with the 46 chromosomes that you inherit from your parents that will determine your growth and development for the rest of your life. So if we didn't go through meiosis, if we just used mitosis to produce your sex cells, what would happen is that your sperm cell would have 46 chromosomes because it went through mitosis. Your egg cell would have 46 chromosomes because it also went through mitosis. Then they would meet up, and all of a sudden, you would have a fertilized egg that has 92 chromosomes in it. And that is not how you make a human. You need 46 chromosomes exactly in order to make a fully functioning human. So to make sure that you only get 1 copy from your mom and 1 copy from your dad, these sex cells have to go through meiosis in order to get created. It turns out some haploid cells. And then when those haploid cells come together, we get what's called a zygote. So a zygote is the very first cell that's produced after fertilization of the sperm and the egg. And when those two come together and fertilize, that is what restores being diploid. That is where you get your 46 total chromosomes from. So that is why meiosis is so important, but only happens in these very specialized cells, is because we need to cut that chromosome number in half so that when the 2 half sets come together, they make a whole set of chromosomes for the newly developing zygote. This zygote is now diploid. It has 2 copies of every chromosome. And now it'll go through millions and millions of rounds of mitosis in order to develop into an embryo, into a baby, into a child, into a teenager, and into finally an adult. So for the most part, the way that meiosis actually works is very similar to mitosis. You get the chromosomes, you get them to line up, you split them apart and form separate cells. But the big difference comes in that in meiosis, you don't just split the cell once. Instead, you go through 2 rounds of division. So during the first round of meiosis, it's very similar to mitosis, where you have your replicated DNA and the homologous chromosomes get separated to either side. So after the first round of meiosis, you have a very similar situation of mitosis, where you have 2 new cells that had their chromosomes, their duplicated DNA separated, and now you have 1 set of DNA in each new cell. One of the interesting things that happens in meiosis, though, is that when these chromosomes all line up down the middle of the cell, they go through that kind of metaphase state, something very peculiar happens, and it's called crossing over. So during meiosis 1, just like with mitosis, you have the homologous chromosomes, and then they're duplicated and they have their chromatids there with them. And all of these chromosomes, these 4 sets of chromosomes, all line up right next to each other in something called a tetrad, because there's 4 of them. And tetra is a prefix that means 4 sometimes. So we have the 2 chromosomes plus their 2 copies all lined up with each other. But when they line up, sometimes something weird happens and they accidentally swap sections of their DNA, of their genes. So it looks something kind of like this. We have our 2 chromosomes, the white one from your mom, let's say, and the blue one from your dad. They duplicate just like they do when you're going through mitosis. So now you've got the 4, the tetrad, all together. But sometimes they accidentally, the ones that are next to each other, can accidentally cross over their DNA strands. It's almost like the DNA gets tangled a little bit, and then when it gets straightened out, they've swapped segments. We won't go super into detail about exactly how this works. It has to do with like the DNA strands themselves mismatching on either side. But essentially the outcome is that now, all of a sudden, you have a slightly different mixture of DNA on your 4 chromosomes. So the 2 that were on the outside are the exact same as before, but the 2 that are on the inside swapped over the DNA. So now a piece of your dad's DNA is on the chromosome that you originally inherited from your mom, and a section of your mom's DNA is on the chromosome that you originally inherited from your dad. So this is what we mean by crossing over. The 2 similar chromosomes, those homologous chromosomes, accidentally swap a small section of their DNA strands. Any questions about meiosis or this crossing over phenomenon so far? So kind of the big takeaway from this crossing over event is that now not every single chromosome has this exact same set of DNA as it started with. You've created a new combination of DNA with these crossed over strands. All right, so we've gone through our first round of meiosis. It's basically looked like mitosis. The chromosomes have been duplicated, they lined up, maybe a little bit of crossing over kind of happens. But then the DNA splits apart and cells split apart. And now we have 2 cells, big whoop. But because it's meiosis, we're not done there. In the second round of meiosis, Meiosis II, we go through the same steps as before, except we don't copy our DNA. So instead, we have the single set of DNA in our cell, and now it gets split in half. So Meiosis II is where we go from being diploid to haploid. After the first division, it's very similar to mitosis. We have a diploid cell we copy the DNA and split it to still get a diploid cell. But then we go through another split, and this is where the DNA and the chromosomes get reduced again by half. So looking at a picture of it kind of really helps to explain it. So we start off with our chromosomes, right? In this case, we have a pair of big chromosomes and a pair of little chromosomes. So this is our diploid normal starting cell. We have a mom and dad big chromosome and a mom and dad small chromosome. DNA replicates just like it would in mitosis. Chromosomes pair up just like they would in mitosis. Maybe a little bit of crossing over happens. So you can see now we have a little bit of blue on this red chromosome and a little bit of red on this blue chromosome. We go through a split. So just like before, we have a pair of big chromosomes and a pair of little chromosomes on each side. But then the big change is when we go through this second split down here. So now not only do both of our cells split again, so now we don't have 2 new cells, we have 4 new cells. And then also, each one of these new cells has half of the amount of DNA as the original cell. So in our original cell up here, we've got 2 big chromosomes and 2 small chromosomes. And our newly created gametes down here, we have one big and 1 little chromosome in each cell. So we've gone from our diploid stage of having 2 copies of each chromosome to our haploid stage of only having 1 copy of each chromosome. 1 big chromosome and 1 little chromosome. Here, this diagram is a little bit less fuzzy, a little bit easier to read. So the first part is all very similar to mitosis again. Again, we have 2 big and 2 little chromosomes in our original cell. The DNA duplicates. They line up to get ready to split. But maybe we get a little bit of crossing over, a little bit of swapping the DNA. Now we go through meiosis I, separate the chromosomes on either side. So still very similar to before, 2 big and 2 little chromosomes in each cell. But we don't stop there. We go through another split. So each one of these new cells gets split again. And now we have 1 copy of each chromosome in our new haploid cells. So 1 big and 1 little chromosome in each one of our new cells. And thanks to that crossing over that happened, now we don't just have pink and blue chromosomes like we did at the start. Now some of our chromosomes are pink with a little bit of blue on it, or blue with a little bit of pink on it. So we switched up the DNA and the genes a little bit thanks to that crossing over that's going on. All right. Any questions about this meiosis process? Why we go through 2 splits, how we end up with the 4 haploid cells? Anything like that, anything that you want me to go back over and talk about some more?
No.
All right, thanks, Paula. Anybody else? Everybody feel okay with the meiosis versus mitosis? All right, so the last thing that I want to mention when it comes to meiosis is the idea of what's called independent assortment. Essentially, this is the idea or the process by which the chromosomes don't always separate the exact same way every time. So it's not like you have your mom chromosome and your dad chromosome, and all of your mom's chromosomes are going to get grouped together, and all of your dad's chromosomes are going to get grouped together. We could potentially pair up any single copy of any chromosome with each other. So what this looks like, for example, is that here we are we're going through meiosis I and meiosis II. And again, we have 2 sets of chromosomes, a big and a little one. And each one has 2 copies, one from the mom and one from the dad. So one possibility is that the way that they line up, both of your dad's copies of chromosomes get pulled to one side, and all of your mom's copies of chromosomes get pulled to the other side. So that then when you split again and create the haploid cell, you could end up where half of your cells contain chromosomes that you directly inherited from your dad, and half of your cells could contain chromosomes that you directly inherited from your mom. So that's sort of this first possibility. Or it's equally likely that instead we pull some of your dad's and some of your mom's chromosomes to one side, and some of your mom's and some of your dad's chromosomes to the other side. So then you end up with a cell where you have some copies of your dad's chromosomes for some sets and some copies of your mom's chromosomes for other sets, and vice versa. So now you end up with a completely different combination of gametes than you did before. Now, some of your cells have your dad's chromosome for, in this case, the big chromosome, and your mom's chromosome for the little chromosome. And then your other cells end up with your mom's big chromosome and your dad's small chromosome. So all this is showing is that your chromosomes don't get evenly matched up and paired away every time. It's completely random. It's completely independent which chromosomes will go with which when the cell divides. And again, you have 46 copies of chromosomes to start with. Half of them are your mom and half of them are your dad. So you could end up with all sorts of different combinations of your mom and your dad's chromosomes being split up and divided into your new gametes, your sex cells. Not only that, but also with the crossing over, that means that in addition to your dad's and mom's chromosomes being separated up at the cell level, they also get mixed up together at the DNA level as well, right? Some of these chromosomes may have mostly your dad's, but a little bit of your mom's DNA on it, and vice versa. So between independent assortment and crossing over, we can get a ton of diversity in terms of the genes that get passed on to the next generation, right? So not every single one of your siblings looks exactly like a 50% mix of your mom and dad, right? And if you have kids, then your kids probably don't look like an exact 50% copy of you and your partner, right? Instead, they're sort of like a blend. Some of them look like they took on more of the dad's traits. Some of them look like they took on more of the mom's traits. Some of them, you're like, how the heck did you turn up like this? You don't look like either one of us. That's all due to all of the different ways that your DNA and your chromosomes can get mixed and matched during this meiosis process. So because the chromosomes get sorted out in different ways when the cell is splitting, and because the chromosomes can mix and match some of the DNA on the chromosomes itself, that means that you can get all sorts of different traits being passed down from parents to offspring. So you do inherit all of your chromosomes from your mom and dad, but each new child, each new offspring inherits a different combination and a different form of those chromosomes. So besides identical twins, which are their whole other separate thing, but besides them, no two offspring from a parent are ever going to look exactly alike or have the exact same chromosome setup. It's all going to be a little bit different, which is good to help there be variety in populations. We want there to be lots of different genes and types of people and organisms out there. It helps populations be nice and healthy. All right. So to kind of summarize what we've been talking about, not just with meiosis, but also kind of comparing between the two in terms of the sort of major events. So some of these we didn't really talk about, so we'll skip over them. But when it comes to things like how many divisions that there are, In mitosis, we just go through the one split, right? We go through prophase, metaphase, anaphase, and telophase once. We replicate our DNA, we split the cell into two identical copies, and that's all she wrote. In meiosis, we go through two splits, two divisions. We go through the entire 4 steps 2 times. We didn't really talk about synapses, so don't worry about that. In mitosis, at the end of it, we have 2 cells. The cells are diploid. They have 2 copies of every chromosome, and they are genetically identical to the parent cell. They have the exact same DNA, the exact same structures, and they're going to do the exact same thing as their parent. In meiosis, on the other hand, we end up with 4 daughter cells at the end of the process, right? We had 2 splits, so 1 cell split into 2, and then those 2 cells split into 4. These cells are haploid, so they only have 1 copy of every chromosome. And they are not the exact same as each other, so each cell gets a little bit different set of chromosomes thanks to the crossing over where we mix and match, and the independent assortment where we mix around how we split up the chromosomes. And they also don't look the same as the parent cell, right? The original cell that we started off with in meiosis was a diploid cell that had the 2 copies, but now all of our daughter cells are haploid, so there's no way they could be the same as the parent cell. Why is it that we go through these processes? Mitosis is kind of used for everyday regulation and repair. So it helps with growth, it helps repair tissue, it helps replace dead cells. So you have skin cells and blood cells that are dying off all the time. They only have a lifespan of anywhere between a couple days to a couple months. So you're constantly having to replenish the skin and blood cells in your body. How do you do this? Mitosis. Obviously, you are a lot bigger now and you have a lot more cells in your body than you did when you were a wee little baby. How is it that you ended up with so many cells in your body? Mitosis. So mitosis is sort of like your normal, average, every day, happens all the time, makes new cells, division. Meiosis is specialized. It only occurs in the sex organs of an organism, and it only is used to produce gametes. So it's only used to make sperm cells or egg cells when we're talking about animal reproduction. So those are the only types of cells that come through meiosis. All of the other cells in your body, muscle cells, brain cells, spine cells, blood cells, skin cells, immune system cells, they all come from mitosis. So mitosis is very general. Meiosis is very, very specialized. All right, any kind of final questions about mitosis, meiosis, the differences between them, anything about either one of those processes that you want me to go back over a little bit more? Alrighty, so I'm assuming since nobody has any questions that you all feel pretty comfortable with all of this, which is great because, actually, let me see. There's a couple other slides that we could talk about, but there aren't really going to be any questions on anything. So we're going to skip over that for now. So your final assignment for this week is basically running through a chart of the differences between mitosis and meiosis. So unfortunately, since this is in a big chart version, I wasn't really able to create it in the Google form. So you'll have to do it on Google Doc or just write down your own, create your own version of this chart with pen and paper. But it basically is a series of questions very similar to the ones that we were just looking at, where you can type in these boxes here, your answers to why do cells do meiosis and why do cells do mitosis? Where does meiosis happen and where does mitosis happen? How many times does the cell divide when we go through mitosis or meiosis? How many new daughter cells are produced at the end of mitosis or meiosis? Do we have any sort of change in chromosomes? And if we do, what kind of change is it? And then what is the final result? When we stand back and look at the process of meiosis and mitosis, what are the final types of cells that we are left with? So again, pretty much exactly what we just spent the last 20 or 30 minutes or so talking about, doing a lot of comparison between these two processes. So hopefully should be pretty simple and straightforward to kind of work through and get turned in. All right, so does anybody have any final questions about the notes, the homework, the midterm, anything that is going on this week they want to ask about before we sign off for the evening?